Welcome to our nocturnal laboratory of gentle discoveries. Here at Dreamtime Science, we transform complex concepts into lullabies for the curious mind. You know what's fascinating? I've been thinking about how the tiniest bits of the universe, the elements, somehow came together to build every living thing around us. There's something remarkable about how simple pieces like carbon and oxygen turned into the incredible complexity of life itself. I can't wait to share with you how this quiet alchemy shaped the very essence of who we are. If these gentle explorations bring you comfort, a quiet like or subscription would mean the world to me. Your support helps keep these peaceful journeys flowing. Take this moment to settle in, wrap yourself in comfort, release the day's weight from your shoulders, and let your breath become as gentle as starlight. Somewhere in the silent velvet of the night, when the world is hushed and the last rays of sunlight have retreated behind the curve of the earth, a different kind of illumination beckons. It is the glimmer of starlight, ancient and indifferent, and yet somehow intimately connected to the story of every living cell. As you lie in the gentle darkness, you might recall the phrase, whispered in science fiction, sung in pop ballads, written across movie posters, "We are made of stardust." It is more than metaphor. It is a truth forged in the furnaces of long-dead stars, a truth that hums within your bones and breathes from your skin. Let your mind drift outward, past the familiar landscapes of forests and rivers, cities and oceans, until you hover in the vast emptiness between worlds. Out here, the sense of scale slips from your grasp. The Milky Way, with its 400 billion stars, is only one among 100 billion galaxies. And yet the atoms that make up your hands, your heart, your thoughts, were all born in the same cosmic ballet that sets these galaxies whirling. Consider the earliest ages of the universe just after the Big Bang, when everything was compressed into a singular, seething point. In those first few minutes, as the universe expanded and cooled, only the simplest of elements could form, hydrogen and helium with a whisper of lithium. The universe was awash with these ghostly gases, but it was a barren expanse, devoid of the heavier elements that would one day build planets, oceans, DNA, and dreams. For those, something more was needed, the fire of stars. In the language of science, this is called nucleosynthesis, the process by which stars forge new elements in their cores. But in the poetry of existence, it is a dance. Think of it as a slow, stately waltz, the partners invisible, the music inaudible, yet the consequences profound. Within the heart of each star, gravity squeezes matter so tightly that atoms are forced to merge. Hydrogen fuses into helium, helium into carbon, carbon into oxygen, neon, magnesium. The rhythm builds, each step a transformation, each note a new element. In the largest stars, this dance continues, forging ever heavier elements, sulfur, silicon, iron, until the core becomes an iron heart, incapable of giving any more energy to the music. And then, in a final crescendo, some of these stars end their lives in spectacular explosions, supernovae, scattering their elemental creations across the universe. Shockwaves ripple through the interstellar medium, stirring clouds of gas and dust. Here in these ephemeral clouds, the seeds of new solar systems are sown. The stardust, carbon, oxygen, iron, calcium, phosphorus, binds together, cooled by the shadows of cosmic night, until gravity gathers it into new worlds. If you have ever gazed at the night sky and felt a strange sense of homesickness, this is why. The calcium in your teeth, the iron in your blood, the phosphorus in your DNA were all forged in stars that died billions of years before the Earth was born. Each atom tells a story older than any myth, a story written in the language of physics and time. Pop culture has always been drawn to this story. In novels and films, stardust is the alchemist's ingredient, the source of magic, the dust that sparkles on the fingertips of cosmic beings. There is a reason for this fascination. The truth is no less wondrous than the fiction. Recall the silver trails of stardust left by the characters in Neil Gaiman's Stardust, or the refrain in Joni Mitchell's Woodstock, where she sings, "We are stardust, we are golden." Science fiction, too, has taken up the theme, from Carl Sagan's reverent proclamations in Cosmos to the swirling nebulae of Interstellar. These stories and songs remind us, sometimes more effectively than textbooks, that we are not apart from the universe, but utterly entangled in its fate. Yet the real stardust, drifting through the void, is nothing like the sparkling powder of fairy tales. It is not so much dust as it is the fine particulate of atoms, carbon grains, microscopic silicates, tiny flecks of iron and nickel, each one a relic of a vanished star. Some of it still floats through our solar system, raining down invisibly onto mountaintops and rooftops, settling in the deep sea, mingling with the dust beneath your bed. If you run your finger along a windowsill and collect a pinch of household dust, a few of those particles will have journeyed across the galaxy, unchanged in the millions of years since they were cast out by their parent star. The very air you breathe tonight contains molecules that have travelled from the farthest reaches of space, swept up by the solar wind, captured in the gravity of Earth. In this sense, every breath is a communion with the cosmos. The oxygen that slips into your lungs was made in the core of a red giant. The carbon that forms your cells was once the fuel of a star's burning heart. Perhaps this is why the motif of stardust recurs in our stories. It is a way of acknowledging a deep ancestral memory, a longing for connection with something vast and beautiful beyond the limits of our brief lives. When we read of heroes forged from star metal or dream of voyages through nebulae, we are remembering in some quiet corner of our minds that the universe is not only our home but our origin. There is comfort in that, a sense that even in the greatest loneliness we are never truly separate from the stars. It is easy to forget in the rush of daily life just how improbable our existence is. The specific arrangement of atoms that forms a living cell, the intricate choreography that allows proteins to fold and DNA to replicate, depends on the presence of just the right elements in just the right proportions. Carbon, with its ability to form complex chains and rings, is the scaffold of life. Oxygen, with its hunger for electrons, powers the reactions that keep cells alive. Iron, nestled in the heart of hemoglobin, carries oxygen to every corner of your body. Trace amounts of other elements, magnesium, calcium, potassium, phosphorus, each play their part. None of these could have arisen in the cold aftermath of the Big Bang. They are gifts from stars. Imagine tracing a single atom of carbon in your body back through time. Before it was part of you, it was part of a plant, perhaps a blade of grass or a leaf on a tree. Before that, it drifted as carbon dioxide in the atmosphere, exhaled by some creature long forgotten. Go back further, and it was bound in the shell of a mollusk, or nestled in the tissues of a dinosaur, or dissolved in the primordial seas. Still further, and you find it as a gas among the dust that spun itself into the sun and planets. And before that, it was part of the core of a star, fused from helium in a process that lasted millions of years. Finally, in a moment of cataclysm, it was hurled into space to wander until it found its way to Earth. Every atom in your body has a story like this, a journey that spans the birth and death of worlds. If you could see the world with the eyes of a physicist, you would know that everything is in motion, everything is transformation. The calcium in your bones is the same calcium that gives strength to coral reefs, the same that glimmers in the shells of ancient trilobites. The iron in your blood once flowed in rivers on Mars, or drifted in the heart of a meteorite. The phosphorus in your DNA is older than the mountains, older than the Earth itself. You are a living mosaic, assembled from stardust, animated by sunlight, shaped by the slow turning of the cosmos. There is a certain humility in this realization, and also a sense of belonging. The universe is not a stranger's house. It is your ancestor, your cradle, your inheritance. When you look at the stars, you are looking at the places where your atoms were born. The night sky is a family album, each point of light a reminder of some forgotten chapter in the story of matter. Science fiction has often played with the idea of stardust as a key to extraordinary powers or knowledge. In the Marvel Universe, cosmic rays and alien elements grant abilities beyond the ordinary. In Arthur C. Clarke's tales, the dust of stars is the substrate for alien life, or the means by which civilizations communicate across eons. Even in more grounded stories, the motif persists, the idea that contact with stardust awakens something within us, a memory of our cosmic origins. But the reality is subtler and no less profound. There is no sudden transformation, no instant revelation. Instead, there is a slow awakening, a deepening of understanding. Every time you learn something new about the universe, you are, in a sense, remembering what you already are. The universe is not outside you, but within you, written in the code of your cells, whispered in the elements of your blood. Even now, the process continues. Stars are born and die, supernovae scatter new elements into space, planets coalesce from the debris. The cycle of creation and destruction is endless, a cosmic dance that has no beginning and no end. We are simply the most recent movement in this symphony, the latest arrangement of stardust, listening to the music of the spheres, and so as you drift into sleep, let your mind wander among the stars. Feel the ancientness of your atoms, the quiet patience of the cosmos. Remember that you are not merely a collection of cells and molecules, but a participant in the grand unfolding of the universe. The stardust that forms you is still dancing, still transforming, still seeking new forms. Somewhere, out in the darkness, a new star is being born, and with it the promise of new stories, new worlds, new lives. Tomorrow we may follow the journey of this stardust as it gathers into planets, oceans, and living things. But tonight, let the thought linger. You and the stars are kin, bound by the oldest dance there is, a dance written in the very fabric of matter, echoing through the silence of space. The music is slow, patient, endless, and you, made of stardust, are part of its song forever. The world of atoms, which we glimpsed as a realm of whirling electrons and nestled nuclei, is not static nor solitary. In the quiet darkness that preceded the dawn of life, these atoms, carbon, hydrogen, oxygen, nitrogen, drifted, spun, and in their own way yearned for connection. The universe, from its earliest moments, has been guided by the invisible hand of attraction and repulsion, the subtle forces that coaxed lonely atoms towards one another, inviting them to join in a dance of exquisite precision and consequence. The tapestry of living matter, the very essence of our own being, is woven from this intricate ballet of bonds. Let us drift for a moment to a time before the first cell, before the first heartbeat, before even the first gentle swirl of a primordial ocean. Here, in the vast and starlit silence, the story of molecular life begins, not with a grand proclamation, but with countless humble encounters. Atoms, each with their own quirks and predilections, approach, hesitate, and sometimes entwine. It is within these unions that something marvelous is forged. Molecules, the true architects of life's endless variety. But what is it that brings these atoms together? The answer lies in the subtle geometry of their electrons. Atoms are not solid marbles, but rather clouds of probability, with electrons whizzing in defined regions called orbitals around their compact nuclei. The outermost layer of these electrons, the valence shell, plays a special role. Nature, ever seeking balance, encourages atoms to fill these shells, for in completeness lies stability. It is as if each atom carries a set of open hands, seeking to clasp those of its neighbors, to share or exchange electrons in pursuit of a more tranquil existence. There are several ways these atomic hands might join. The first, and perhaps the most poetic, is the covalent bond. In this arrangement, two atoms share one or more pairs of electrons, holding each other in a gentle but unyielding embrace. Consider, for instance, two hydrogen atoms. Each alone possesses a single electron, one shy of the coveted pair that would fill their outermost shell. As they draw close, their electrons begin to dance between the two nuclei, a duet that binds them as a molecule of hydrogen gas, H2. The electrons are not torn from one atom to the other. Rather, they are shared, like a pair of hands clasped tightly in mutual support. Carbon, the star of life's molecular cast, is a master of covalent bonding. With 4 open spots in its valence shell, it can form up to 4 bonds, arranging them in the shape of a perfect tetrahedron. This symmetry is not mere aesthetic. It allows carbon to link with other atoms in endless diversity, forming chains, rings, and branching networks that are the backbone of organic molecules. Methane, the simplest of these, is a single carbon atom embraced by 4 hydrogens, each sharing a pair of electrons. But as more carbons join the dance, the possibilities multiply, giving rise to the elaborate frameworks of sugars, fats, and proteins. Another form of atomic union is the ionic bond, a more forceful arrangement born of unequal partnerships. In this case, one atom donates an electron completely, while another eagerly accepts it, and the resulting ions, charged particles, cling together by the pull of opposite charges. Sodium and chlorine, for example, are unlikely partners on their own. Sodium, with one lonely electron in its outer shell, is eager to give it up. Chlorine, with 7, longs for just one more. When they meet, sodium surrenders its electron to chlorine, becoming a positively charged ion, while chlorine, now with a full shell, becomes negatively charged. The two are drawn together by the electric force, forming the familiar crystal lattice of table salt, NaCl. It is a bond not of sharing, but of mutual need and electrostatic embrace. Yet in the grand theatre of biology, it is the subtler, more flexible bonds that take center stage. Covalent bonds may form the sturdy walls and pillars of molecular architecture, but life itself depends upon the ability of molecules to interact, to touch and separate, to form fleeting associations and delicate partnerships. For this, we turn to the world of weak bonds, hydrogen bonds, and van der Waals forces, whose very fragility is their strength. Consider water, the matrix of life. Each molecule consists of 2 hydrogens covalently bonded to an oxygen, arranged not in a straight line, but in a bent, angular configuration. This shape, combined with the differing electronegativities of oxygen and hydrogen, means that water is a polar molecule. One end bears a slight negative charge, the other a slight positive. When water molecules approach one another, the positive region of one is attracted to the negative region of another, forming a hydrogen bond, a tenuous link, easily broken and easily reformed. It is these hydrogen bonds that confer upon water its remarkable properties, its high boiling point, its ability to dissolve a vast array of substances, its expansion upon freezing. But hydrogen bonds are not unique to water. Within the spiraling ladder of DNA, they form the rungs, pairing the bases adenine with thymine, guanine with cytosine, in a code that underpins the very instructions for life. These bonds are weak enough to be separated, essential for the copying and reading of genetic information, yet strong enough to hold the double helix together. In proteins, too, hydrogen bonds guide the folding of chains of amino acids into the intricate shapes necessary for function. Even weaker than hydrogen bonds are van der Waals forces, subtle attractions arising not from permanent charges, but from the temporary fluctuations in electron clouds. Imagine two molecules, each minding its own business, drifting close enough that their electron clouds begin to influence one another. For a fleeting moment, a region of negative charge may appear, inducing a corresponding region of positive charge in its partner, and the two are drawn together by this ephemeral dance. Though van der Waals forces are faint, they become significant when many atoms are involved, as in the folding of proteins or the stacking of lipid molecules in a membrane. They are the whispers and gentle nudges that together shape the grand symphony of life. It is tempting, perhaps, to imagine chemical bonds as rigid, deterministic structures, like the beams and girders of a machine or the links of a chain. But the reality is more subtle, more dynamic. Bonds are not static. They vibrate, twist, and sometimes break, only to reform in new configurations. Molecules are in constant motion, jostling and colliding in the warm bath of the cell. Chemical reactions are not singular, dramatic events, but rather the consequence of billions upon billions of such encounters, each governed by the probabilities of quantum mechanics and the thermodynamic hunger for lower energy and greater entropy. A common misconception is that molecules are fixed entities, their shapes and compositions immutable. But in truth, the molecular world is alive with motion and transformation. Enzymes, remarkable molecular machines fashioned from proteins, catalyze reactions by bringing specific atoms into just the right orientation, lowering the barriers to bond formation or breakage. Without enzymes, the chemistry of life would proceed so slowly as to be effectively impossible. With them, the dance of molecules becomes a frenzied waltz, each step choreographed by evolution to suit the needs of the cell. And so, from the smallest ions to the largest macromolecules, the story of life is written in the language of chemical bonds. Sugars, with their intricate rings and branching chains, store the energy of sunlight, ready to be released in the controlled burn of metabolism. Fats, composed of long chains of carbon and hydrogen, cluster together in water, forming membranes that both contain and protect. Proteins, with their dizzying array of forms, execute the myriad functions of the cell, catalyzing reactions, ferrying molecules, transmitting signals, providing structure. Nucleic acids, built from just a handful of building blocks, store and transmit the instructions for all of this marvelous machinery. Each of these molecules is assembled from simple elements: carbon, hydrogen, oxygen, nitrogen, phosphorus, sulfur, joined by the elegant logic of chemical bonding. Yet, the complexity that emerges from these unions is astonishing. The difference between a droplet of oil and a strand of DNA, between a grain of salt and a beating heart, lies not in the elements themselves, but in the patterns of their assembly. Nature, through countless generations, has explored the possibilities, selecting and refining those combinations that best capture energy, store information, and replicate themselves. There is another misconception that lingers in popular imagination, that chemical reactions are akin to miniature explosions, violent and unpredictable. While some reactions do release energy in dramatic fashion, most of life's chemistry is far more subtle, an endless series of small steps, each carefully regulated, each proceeding only when conditions are just right. The cell is not a cauldron of chaos, but a meticulously orchestrated theatre where molecules meet, embrace, and part in a continuous performance. It is also easy to suppose that molecules are isolated, acting alone within the watery confines of the cell. Yet, the truth is one of community and context. Molecules interact not just with each other, but with the surrounding environment: water, ions, other macromolecules, all influencing the outcome of each reaction. The folding of a protein, for instance, is not dictated solely by its own sequence, but by the push and pull of the solvent, the presence of other molecules, the delicate balance of forces within the crowded cytoplasm. Sometimes bonds are broken not by the will of the molecules themselves, but by the intervention of energy, heat, light, or chemical input. In photosynthesis, for example, photons from the sun strike the chlorophyll molecules in a plant's leaf, exciting electrons and triggering the formation of new bonds that capture solar energy in chemical form. In respiration, the bonds within glucose are broken step by step, releasing the stored energy to power the myriad activities of the cell. At every scale, from the invisible intimacy of atoms to the grand architecture of living beings, the story is one of connection. Bonds form and break, molecules assemble and disassemble, always in flux, always moving towards new possibilities. The ballet is not choreographed by any conscious hand, but emerges from the fundamental laws of physics and chemistry, a testament to the creative power of nature expressed in the language of atoms and molecules. As we drift deeper into the molecular heart of life, it becomes clear that the beauty and complexity we observe are not the result of magic or accident, but of innumerable interactions, each governed by the simple yet profound rules of bonding. It is in this hidden symphony that the potential for life was born, waiting only for the right conditions, the right arrangements, to burst forth into self-replication and evolution. The ballet continues, unseen but unceasing, in every drop of water, every breath of air, every living cell. The bonds that form and break, that twist and reform, are the silent music that animates the world. And as we settle deeper into the night, the question lingers: how did these molecules, assembled from atoms in the depths of space and time, first come together to form the earliest stirrings of life? What pathways led from simple matter to the first self-replicating molecules, the first steps on the long journey to consciousness? The answer lies beyond the mere formation of bonds, beyond the architecture of molecules themselves, within the dynamic chemistry of early Earth, where energy and matter mingled in 1,000 different ways, seeking the spark that would ignite the story of living things. The intricate ballet of bonds is only the beginning. It is within the restless transformations of chemistry that life's true magic would emerge, waiting to be discovered in the next unfolding act. If you listen closely, almost as if tuning your ears to the hush of midnight rain, you can detect the faint, intricate music of life itself, a melody composed not of notes, but of elements. It is an ancient song, woven from carbon, oxygen, hydrogen, nitrogen, sulfur, and phosphorus, with a scattering of metals and mysterious trace elements, all dancing in unending patterns. But to truly appreciate this symphony, to understand how the elements of the universe are coaxed into the forms of birds, trees, and ourselves, one must learn to watch nature at work in her most secret laboratory, the living cell. For countless generations, these workings remained hidden, their details shrouded in a veil as fine as the mist that settles gently upon a meadow at dawn. The ancients saw only the surface, a leaf trembling in the wind, a child's laughter, the quicksilver flash of a fish below a pond's surface. Yet the true marvels, the alchemy of life, unfolded invisibly within. It would take centuries, a slow accretion of curiosity and ingenuity, for humanity to develop the tools to peer into these depths. The journey began with the simplest of lenses. In the 17th century, Antonio van Leeuwenhoek, a Dutch tradesman with a passion for grinding glass, pressed his eye to a tiny, handcrafted microscope and glimpsed a world no one had seen before. There, in a drop of water, he found animalcules, tiny living beings bustling with purpose. His letters to the Royal Society in London echoed with wonder, describing creatures smaller than the width of a hair. Yet even Leeuwenhoek, with his singular vision, could not have guessed at the elemental theatre within each of those minuscule beings. As centuries turned, the microscope grew in power and sophistication, and the secrets of nature's laboratory began to yield. Scientists stained cells with dyes, observing how the colors clung to certain structures. They noticed the nuclei, the mitochondria, the intricate networks of membranes. But what were these structures made of? What elemental threads wove the fabric of life? To answer that, researchers borrowed tools from the chemist's arsenal. The cell, after all, is not merely a bag of fluid, but a living crucible where elements are combined, broken, and recombined with exquisite precision. Early investigators, armed with flasks and crucibles, ground up tissues and bones, burned them to ash, and analyzed the residues. They found carbon in abundance, the hallmark of organic matter, phosphorus in bones and brains, iron in blood. These were the first hints that life's diversity was built from a surprisingly small palette of elements. But to truly decode nature's laboratory, scientists needed to see not just what was present, but how these elements interacted, how they were assembled into molecules, how those molecules moved and changed, how they made the difference between the living and the dead. This challenge called forth a new generation of experimentalists whose ingenuity would open windows into the living world's most private chambers. One of the most revolutionary of these experiments unfolded quietly in a laboratory in Uppsala, Sweden, in the early 20th century. The biochemist Theodor Svedberg, fascinated by the mysterious particles found in egg yolk and blood, devised a machine that would become legendary, the ultracentrifuge. With it, he could spin samples at dizzying speeds, subjecting them to forces thousands of times greater than gravity. The result was a silent, relentless sorting of molecules by their size and density. Proteins, nucleic acids, and other cellular components, once lost in a molecular soup, now separated into distinct layers, each a clue to the underlying architecture of life. With the ultracentrifuge, Svedberg and his contemporaries began to unpick the tangled web of life's chemistry. They saw that proteins, those miraculous machines within our cells, are built from chains of amino acids, each one a unique arrangement of carbon, nitrogen, hydrogen, oxygen, and sometimes sulfur. They glimpsed the nucleic acids, long, elegant molecules carrying the genetic code, their backbones laced with phosphorus. But how were these elements, so simple in isolation, coaxed into such complexity? The answer lay in the subtle forces of chemistry, the bonds that form when elements share or exchange electrons, the intricate shapes they twist themselves into, the way each molecule fits like a key into the lock of another. Here, scientists turned to the growing power of X-ray crystallography, a technique that would allow them to see the arrangement of atoms within a molecule by studying the patterns produced when X-rays scattered through a crystal. It was with this technique that Dorothy Crowfoot Hodgkin, in the 1930s and 40s, first glimpsed the three-dimensional structures of complex biological molecules. She revealed the atomic architecture of penicillin, of vitamin B12, and later of insulin. Each structure was a revelation, a map of the ways in which carbon chains curled, the places where nitrogen and sulfur nestled in, the phosphates that linked together like beads on a string. Yet the most profound leap in decoding nature's laboratory came with the unraveling of the double helix. In the early 1950s, James Watson and Francis Crick, building on the meticulous X-ray images taken by Rosalind Franklin and Maurice Wilkins, proposed a model for the structure of DNA. Here, at last, was the blueprint of life itself, two intertwined strands of nucleotides, each containing a sugar, a phosphate, and a base made of nitrogen and carbon. The bases, adenine, thymine, guanine, cytosine, paired up in precise arrangements, guided by hydrogen bonds as delicate as a spider's silk. In this elegant spiral, the instructions for building every living thing were encoded, written in a language of elements. But the study of life's chemistry is not confined to the molecules alone. It extends to the flow of elements through living systems, the cycles of carbon and nitrogen, the balance of minerals, the dance of ions across membranes. To study these processes, scientists have devised ingenious ways to follow the path of individual atoms as they move through the labyrinth of life. One of the most powerful tools for this purpose is the use of isotopic tracers. Isotopes are atoms of the same element that differ slightly in mass, owing to their differing numbers of neutrons. Some, like carbon-14 or phosphorus-32, emit a faint radioactivity, a whisper that can be detected with sensitive instruments. By introducing these isotopes into living organisms, scientists can watch as the labeled atoms are absorbed, transformed, and incorporated into new molecules. In the 1940s and 50s, this technique was used to great effect by Melvin Calvin and his colleagues, who sought to unravel the secret of how plants transform sunlight into food. By feeding algae with carbon dioxide containing radioactive carbon-14 and then quickly killing the cells at various intervals, Calvin's team could track the journey of the carbon atom through a complex web of chemical reactions. They discovered the Calvin cycle, the sequence by which plants fix carbon, transforming it into sugars that form the foundation of the food chain. Here again we see nature's laboratory at work, taking simple elements and weaving them into the intricate patterns of life. Elsewhere, scientists have used isotopic tracers to probe the mysteries of metabolism, how our bodies harvest energy from food, how nitrogen is recycled in the soil, how calcium moves through bones. In medicine, radioactive isotopes serve as diagnostic tools, illuminating the flow of blood, the activity of the thyroid, the spread of cancer. Each experiment is like a firefly's glow in the darkness, a fleeting illumination of the pathways along which life's elements travel. But the tools of nature's laboratory are not limited to the physical sciences. With the dawn of the molecular era, a new set of instruments emerged, ones that could probe the very code of life. The polymerase chain reaction, or PCR, devised by Kary Mullis in the 1980s, allowed scientists to amplify tiny fragments of DNA, making visible the genetic instructions that were once hidden deep within a cell. In the hands of geneticists and biochemists, this technique unlocked the secrets of heredity, disease, and evolutionary change. To peer ever deeper, researchers have turned to the electron microscope, which sends beams of electrons far smaller than any photon through thin slices of tissue, revealing structures at the scale of nanometers. Here we see the ribosome, a molecular machine built of proteins and RNA, orchestrating the assembly of new proteins atom by atom. We see the channels in membranes, tiny gates that allow sodium, potassium, calcium, and chloride ions to move in and out of cells, triggering the signals that underlie thought, sensation, and movement. Still, even the most powerful microscopes and incisive chemical techniques would be blind without the guiding hand of experiment. Throughout history, it is the well-chosen experiment that deciphers nature's code, that lays bare the principles by which elements interact in living things. Among the most elegant of these experiments was the work of Frederick Griffith in 1928, who studied the bacteria that cause pneumonia. He observed that a harmless strain could be transformed into a deadly one by exposure to the remains of dead, virulent bacteria, a mysterious transforming principle at work. Years later, Oswald Avery, Colin MacLeod, and Macklin McCarty showed that this principle was DNA itself, not protein as many had assumed. In their careful, painstaking experiments, they isolated DNA, showed it could transform harmless bacteria, and in doing so, proved that the genetic instructions for life were written in this slender molecule. With each discovery, scientists have refined their understanding of how the elements interact within living things. They have learned that enzymes, those marvelous biological catalysts, work by orienting atoms just so, lowering the barriers to chemical reactions and bringing about transformations that would otherwise be impossible in the conditions of life. They have found that metals, though present in trace amounts, play vital roles. Iron at the heart of hemoglobin, shuttling oxygen through our blood. Magnesium nestled at the core of chlorophyll, capturing sunlight in leaves. Zinc stabilizing the folds of proteins and mediating the signals that pass from neuron to neuron. The laboratory of nature is not a sterile chamber, but a dynamic, ever-changing environment. Within each cell, elements are in constant flux, carbon dioxide in, oxygen out, ions pumped ceaselessly across membranes. The machinery of life is maintained by gradients, differences in concentration, in charge, in energy. These gradients drive the processes that sustain us, from the beating of our hearts to the transmission of thoughts across the synapses of our brains. To observe these processes in real time, scientists have developed fluorescent markers, molecules that glow under certain wavelengths of light. By attaching these markers to proteins, DNA, or ions, researchers can watch as molecules move, interact, and change within living cells. The cell, once a black box, is now illuminated from within, its workings visible in bursts of color and light. Yet for all our ingenuity, there remain mysteries unsolved, processes only dimly glimpsed, interactions that elude even our most refined instruments. The complexity of life is such that each answer births new questions, each experiment opens new avenues of inquiry. Nature's laboratory, it seems, is inexhaustible in her capacity for surprise. Tonight, as you drift in the space between waking and sleep, let your mind wander through these hidden workshops, where elements are shaped and reshaped in endless variation. Imagine the patient work of scientists peering through microscopes and spectrometers, coaxing secrets from the silence of the cell. Consider the experiments yet to be designed, the tools yet to be imagined, the discoveries that await in the shadows just beyond our reach. For even as we grow ever more adept at decoding nature's laboratory, we are reminded that the story is far from complete. There are frontiers we have not yet crossed, questions that tremble on the edge of understanding. And so, in the gentle hush of night, we pause, listening for the next note in the endless song of life, still unfolding, still mysterious, still to be explored. Let us rest now at the quiet threshold where the journey turns inward, where the elemental voyage we have traced through the cosmos, the earth, and the intricate machinery of living cells finally circles back to the heart of our own being. The human form, so familiar, so intimately ours, stands as a living testament to the grand, unbroken story of the elements. Tonight, we linger in this reflective chamber and dwell on the meaning that arises from knowing, truly knowing, that our bodies are mosaics assembled from the very dust and fire of the universe. To recognize oneself as elemental is to see with new eyes, to apprehend that our flesh, our breath, our thoughts are not isolated miracles, but rather the flowering of ancient processes. Each element within us is a thread in a tapestry woven over eons, from the earliest starlight to the present moment. Consider the atoms in your hand, the carbon that forms the pattern of your skin, the calcium that shapes your bones, the oxygen carried in every inhalation. These atoms are not new, they are, in a sense, immortal. They have belonged to stars, stones, oceans, and other living beings, a ceaseless migration, a pageant of transformation. The elemental composition of a human, nearly 2/3 oxygen, a fifth carbon, a 10th hydrogen, with nitrogen, calcium, phosphorus, and a scattering of metals and trace elements, reads like a recipe. Yet it is more than a list of ingredients. Each element carries a story, a memory of where it has been, what it has become, and what it might yet do. In the marrow of your bones, calcium atoms once fused in the heart of an ancient star. The iron in your blood, crimson and vital, was forged in the catastrophic death throes of a supernova. The water you drink and the oxygen you breathe are cycled through the biosphere, shared endlessly among all living things. To be human is to be a confluence, a crossroads, a gathering place for the universe's matter. This elemental perspective, at first so vast and humbling, also brings a strange intimacy. If you follow the path of any atom in your body backward, you find yourself moving not only through the history of the Earth, but through the wider narrative of the universe. The hydrogen in your cells is the most ancient, born in the first minutes after the Big Bang, when the universe was but a roiling plasma of particles and light. That hydrogen, enduring and ubiquitous, has drifted across billions of years, perhaps passing through clouds of interstellar gas, being swept up in the formation of the solar system, raining down as part of water on the early Earth, and eventually joining the chemistry of life. Every time you drink a glass of water, you are partaking in this lineage, quenching your thirst with a substance as old as time itself. Oxygen, the most abundant element in your body, comes next in this temporal hierarchy. It was not present in the newborn universe, but was forged in the hearts of massive stars, scattered across the cosmos by supernovae and incorporated into planets, rocks, and oceans. On Earth, oxygen became the great liberator, fueling combustion and respiration, driving the metabolism of every animal, including ourselves. The oxygen you breathe tonight may have been part of a green leaf last week, a raindrop last year, a glacier 1,000 years ago. It is forever on the move, forever entering and departing the realm of life. Carbon, meanwhile, is the backbone of biology, the element around which the architecture of living molecules is built. Its story is one of flexibility and possibility. With 4 valence electrons, carbon can form stable bonds in endless configurations, allowing for the intricate branching and folding of organic molecules, proteins, nucleic acids, sugars, fats. The carbon in your body, too, has a deep history. It is stardust, recycled through countless cycles of life and death, photosynthesis and respiration. When you exhale, you return a fraction of this carbon to the atmosphere, where it may one day be drawn down into the roots of a tree or the shell of a mollusk. The dance of carbon is the dance of life itself. Even the trace elements in your body, present in quantities so small you might think them trivial, are essential and storied. Zinc, copper, selenium, iodine, molybdenum, manganese, each plays a subtle, precise role in your biochemistry. A zinc atom, nestled at the heart of an enzyme, may have spent millions of years locked in a vein of ore before being liberated by weathering, taken up by a plant, and finally finding its way into your cells. The selenium that fine-tunes your antioxidant defenses may have journeyed from deep within the Earth's crust, carried by rivers and absorbed by crops. Even the rarest elements are woven into the living fabric. Yet for all this grandeur, it is easy to forget the sheer ordinariness of these materials. The elements are everywhere, lying silent in rocks and air, in rivers and dust. What makes a human being, what makes life so extraordinary, is not the rarity of our substances, but the pattern in which they are arranged. There is no difference, chemically, between the oxygen in your lungs and the oxygen in the ocean, between the iron in your blood and the iron in a meteorite. It is the choreography, the organization, the living architecture that distinguishes us. We are not made of special matter, but we are made in a special way. This realization can be at once humbling and elevating. On the one hand, it reminds us that we are not separate from the world, not immune to its laws or distinct from its materials. We are fundamentally of the earth. Every cell, every breath, every thought binds us to the rocks and rivers, the winds and the stars. The processes that build and sustain our bodies are the same as those that shape the mountains and seas. We are, as Carl Sagan so famously put it, star stuff, but we are also earth stuff, water stuff, air stuff. We are the sum of the planet's history and the universe's creative fire. On the other hand, to look closely at the arrangement of these elements is to see the emergence of something new. Out of the simple interactions of atoms arise the complexity of life, the metabolism that fuels each heartbeat, the neural networks that give rise to mind and memory, the consciousness that allows us to ponder our own origins. This is the mystery at the heart of the elemental human, how matter, so ancient and indifferent, can give rise to thought, to love, to curiosity. How, from the dance of electrons and the folding of proteins, emerges the experience of being alive. There is a kind of poetry in this, a resonance between the outer and the inner, the cosmic and the personal. When you touch your skin, you are touching the universe. When you look into the eyes of another, you are seeing stardust made self-aware. The boundaries between self and world begin to blur, and what remains is a sense of participation, of belonging. This, perhaps, is what the study of our elemental nature ultimately offers, a sense of kinship, not only with other living beings, but with the whole of existence. Let us dwell for a moment on what it means to be a gathering of elements in this particular age. The atoms in our bodies are ancient, but the configuration they take is fleeting. The human form, with its upright posture, nimble hands, and ceaseless mind, is the result of a long evolutionary journey. Each adaptation, each branching of the tree of life, has been shaped by the availability and behavior of elements. The rise of multicellular life depended on oxygen's abundance. The emergence of bones required calcium. Nerves and brains flourish thanks to sodium, potassium, and phosphorus. The world's chemistry has guided the course of life, and life, in turn, has transformed the chemistry of the world. The oxygen-rich atmosphere we breathe is the legacy of ancient cyanobacteria. The carbon locked in fossil fuels was once sunlight captured by plants. The cycles of elements through land, sea, and sky are interwoven with the history of life itself, and here we find another layer of meaning. To know the elements within us is to understand our dependence on the wider web of life and the planet's great cycles. We do not create the elements, we borrow them, use them, and return them. The food we eat, the air we breathe, the water we drink, these are not possessions, but gifts, part of a perpetual exchange. Our bodies are temporary assemblies, brief gatherings of matter that will one day disperse. The calcium in your bones may one day rest in a seashell. The nitrogen in your muscles may nourish a patch of grass. The elements move on, indifferent to our passing, yet marked by our presence. There is, in this, a kind of solace. The fear of mortality, so universal and so deeply felt, softens when one remembers that nothing is ever truly lost. The elements of your body have been many things before you, and will be many things after. Death is not an end, but a transformation, a return to the great cycle. The boundaries of self dissolve, and what remains is the continuity of matter, the persistence of the elements. Yet this continuity brings responsibility. To recognize oneself as elemental is to see the world with new eyes, to realize that the health of the planet is inseparable from the health of our own bodies. The pollution of air and water, the depletion of soil, the alteration of the climate, all these are disruptions of the elemental cycles on which life depends. The oxygen that fills your lungs may carry the trace of distant fires. The mercury in your nervous system may have travelled through rivers and fish. Our actions, large and small, ripple through the web of matter, affecting not only ourselves, but the generations to come. The story of the elemental human is, in this way, both ancient and urgent. It tells us where we have come from, and hints at where we might go. It is a story written in the language of atoms, in the patterns of molecules, in the cycles of the earth and the stars. It is a story we share with every living thing and with the planet itself. As you lie in quiet reflection, feel the pulse of your heart, the rhythm of your breath, the subtle warmth of your skin. Know that within you the universe is alive and aware. The hydrogen in your blood has seen the dawn of time. The calcium in your bones has weathered the collapse of stars. The carbon in your brain has cycled through countless lives. You are, in every sense, elemental, a fleeting configuration of ancient matter, a brief spark in the long night, a witness to the unfolding of existence. And so, as the night deepens and your thoughts grow quiet, let the wonder of your own elemental nature settle upon you. There is much yet to ponder, much to explore. The journey of the elements is far from over. The atoms in your body will continue their voyage long after your own story has come to a close. The universe in its ceaseless transformation will find new ways to arrange its matter, new forms in which to awaken. In every breath, in every heartbeat, you are part of this cosmic unfolding, a participant in the great dance of the elements that has no end. As our journey through the alchemy of life gently drifts to a close, we watch the elements weave their cosmic dance, like a comet tail fading softly into the hush of space. May this quiet wonder cradle your thoughts tonight, inviting peaceful dreams of stardust and life's mysterious spark. Return soon, dear dreamer, for more tales whispered from the depths of the universe.